I'd also like to thank Dr. Abdenur for co-hosting the event and approving us to do the event. I'd also like to congratulate Dr. Abdenur on being the director of CHOC UCI Center of Excellence. So huge congrats to him. He's the chief Division of Metabolic Disorders and the director of the Metabolic Laboratory at CHOC. CHOC and the University of California, Irvine have been jointly designated a National Organization for Rare Disorders, or NORD, Rare Disease Center of Excellence, joining a new and highly select group of 31 medical centers seeking to expand access and advance care and research for rare disease patients in the United States. Dr. Abdenur will serve as the director of the NORD Center of Excellence Partnership. So thank you so much, Dr. Abdenur, and I will go ahead and turn the floor over to you.
Well, thank you so much for your presentation. I want to thank the organizers. I think this so far has been a wonderful event, and I know how much work is behind it. The fact that CHOC, not particularly me, is hosting this event is because we are definitely committed and interested in helping families dealing with rare disorders, and as much as possible, we will offer our platform and support for them to share information and good news about science. So let me see if I can advance. For those who don't know who we are or where are we, as there are people from everywhere, on my right upper right is the map of the US, and I'm sure you are familiar where California is. On the left side, California is a huge state with more than 40 million people by now living there. So we are in a tiny place of California down here in what we call Orange County, but even though it's a small county in terms of extension, we have more than 3 million people living here, like a small European country probably, and we have approximately 40,000 newborns a year. On top of that, our hospital, being a quaternary children's hospital, takes care of children coming from neighboring counties, Riverside and San Bernardino. So we really receive the children who are with the more severe and rare disorders. And that is why over the last almost 20 years, I've been trying to develop what we call a center for rare diseases. And this is what we have so far, what we call our metabolic rare disease program, where we have a combination of clinical services, basic science with bench research, the possibility of screening for different drugs and creating animal models with the idea of going to clinical trials and experiments in patients. We do not have a diagnostic laboratory specific for mitochondrial diseases. We do have what we call a general metabolic laboratory where we do the initial testing that may help suspect a mitochondrial disease, but we don't do the specific genetic or post-genetic analysis to identify mitochondrial disorders. Our patients come to one of three clinics, either the lysosomal clinic, where we have a big variety of patients, our neurometabolic clinic, where we see the mitochondrial patients, and it's a multidisciplinary clinic. And then we have a clinic for other inborn metabolic diseases, where we see, again, a variety of conditions. Those clinical surveys are only possible because we have a multidisciplinary approach where the family is at the center, and we have a team not only of physicians, nurse practitioners, but also dietitians, nurses, case managers, social worker, interpreter, and more important, very important, genetic counselors. And that allows us to take care of the full family. We know how difficult these diseases are. Many families have expressed how long time it takes for the diagnosis, how difficult it is to go from one clinic to another, from one specialty to another. So we try to facilitate that in the multidisciplinary environment. And over the years, in our metabolic center, we have either diagnosed or received or been referred patients with many, many different conditions. This is just an overview. We have all metabolic pathways that you can imagine of amino acid disorders, fatty acid oxidation defects, etc. And highlight here are more than probably 60 well-identified and diagnosed mitochondrial diseases. And I know different laboratories specialize in different mitochondrial conditions. So we put together a slide where we have more or less a breakdown of the different conditions we have diagnosed and are taking care of, or we took care of, including affecting mitochondrial DNA or nuclear DNA. And of interest for this group today, these are the mitochondrial protein synthesis defects that we have diagnosed or we are taking care of. I put TFAM in here in the synthesis section, but I saw Dr. Rita Hobart putting TFAM in the group of protein synthesis. So I'm glad you did that because we described so far one patient with TFAM and was a very interesting story. We also, just for those researchers who may be interested, are very focused on two genes that are not mitochondrial itself by themselves, but they affect mitochondrial pathways. And those are the HIVCH and ECHS1 genes that affect the Valine catabolic pathway. So as Dr. Graneski was mentioning before at UPenn, when we have clinical questions, we have a very open bed to bench approach. So we go from the clinic to the basic science laboratories. And in this particular case for mitochondrial diseases, we go to the laboratory for energy metabolism. And this is a laboratory that we humbly established a few years ago. And we try to investigate the function of mitochondrial genes. We try to understand how genetic defects affect different metabolic pathways. We study potential treatments in vitro, and the goal is, of course, the dream, I would say, of everybody to be able to develop clinical trials. And all these aims or goals are not different from probably each and every laboratory that has been presenting so far. So I will also tell you the story of a family who we diagnosed and prompted our interest and research in this particular gene. As the other physicians presenting before and researchers, many times your research pathway is not defined by your particular previous background, but by a particular family that motivates and inspires you to work. In this particular case, it's about leukoencephalopathy with brainstem and spinal cord involvement and lactate elevation, the condition that Dr. Fatimi early on referred to and was beautifully described by the Dutch group many years ago. As we already saw, DARS2 encodes for aspartyl tRNA synthetase. And children or adults can be affected. There is a heterogeneous clinical presentation. Onset in infancy will result in a very severe disease. Onset could be in adults or adolescents with a mild and slowly progressive disorder, which is more common. And finally, there are some cases reported with induced paroxysmal ataxia. There is a very characteristic MRI pattern that was also described by the Dr. Marc van der Nap many years ago. And then we know now that the genotype appears to somehow influence the phenotype. That is a common theme that we have seen in many of the presentations today. So there is not just one particular disease, there is one particular disease and one particular genotype that affects how the disease is expressed. So accurate genetic diagnosis is really essential. This is our initial patient. He was the product of a triplet pregnancy, and this is the index case here. Because of the triplet pregnancy, he was born premature at 28 weeks and had a, I would say, rather typical course in the NICU, staying for about 3 months with some minimal complications, but he fully recovered and was discharged home and was doing very well, thriving together with the other 2 siblings. The parents reported a history of intermittent headaches since early age, about 5 years of age, that was investigated, but no clear diagnosis was established at the time. And when he was 8, the headaches were worsening, and an MRI was ordered by a neurologist, and it was abnormal, was suggestive of a leukodystrophy, and then he was referred to us. Besides the triplet, there was another sibling, a boy who was born a few years later. This is our initial evaluation, and he at 8 years of age was absolutely normal and continues to be absolutely normal. Biochemical testing was non-revealing, as it happens in many of these disorders. That is, in this particular condition, no elevated lactic acid or abnormal organic acid or amino acids. The molecular testing, we did a gene panel at that time initially showed 1 mutation that had been previously reported in intron 5 of the DARS2 gene. And based on 1 mutation and the abnormal MRI with hyperintensities in the supratentorial white matter that were partially confluent, and also the sparing of the U fibers, plus some other abnormal intensities in the medulla, in the pons, and in the upper cervical spine, it was really suspected that the real gene was indeed DARS2, even though we didn't have the second mutation. So here is a beautiful graph from the Dutch group published by Van der Berg, I believe. And here the red arrow shows the mutation that was identified by the commercial laboratory in intron 5. So we had to look for the second mutation, and we know there is a hot spot for mutations, usually leaky or mild mutations in intron 2 that had not been covered by the laboratory that performed the initial testing. So we went to the laboratory, and in our e-lab, we Sanger sequenced that region of the gene, and we were able to uncover the second mutation that, even though it's in that region, it hadn't been reported before. And again, it's a splice mild mutation. We were also able to stable the inheritance pattern, detecting 1 mutation in the mother and the father that explained the 2 mutations in the proband. The family was very eager for answer, very willing to cooperate and collaborate in more studies and research. So we went ahead and studied all family members, especially because some children may be completely asymptomatic. And we unfortunately identified a second affected sibling, the youngest brother, who had exactly the same mutation pattern as the proband. Now, there's always a thin layer that silver layer that we can take, and we found that sibling 1 in this family did not harbor any of the 2 mutations in the DARS2 gene, and therefore became our normal control for all the studies that I'm going to show. It's good, as we discussed before with other researchers, to have a control that is similar in the genetic background to your own patients, and in this case, we couldn't ask for a more perfect control. This is just to show that the younger boy who had been identified because of the mutation analysis was completely asymptomatic, but had a history of intermittent leg pain, which we are not sure is related to the disease, even after following him for a while. And at age 5, when he was able to tolerate an MRI without anesthesia, there were already white matter abnormalities of, again, confluent hyperintensities in T2 and sparing the U fibers, an accidental finding of an intraventricular cyst. So we consider patient 2 also affected with the same disease, as he had the same genotype and phenotype. So with all this information, again, with a family eager to collaborate, we obtained skin biopsies from all family members and proceeded to do some studies. And the first thing we did was to do RT-PCR, looking at the possibility of the skipping of the exon 3 in the patients. And as you can see, you expect a higher product or transcript when you have all the exons. In this particular case, we use primers in exon 2 and exon 4. And then if you skip the exon 3, the size of your product will be smaller. And you only see that in the patient 2, patient 1, and the father. You don't see that in the heterozygous siblings, except for sibling 2, who is a carrier of the father mutation. And you don't see it in the mother. The second thing we did is to quantitate the DARS2 mRNA levels. And for that, we use a normal adult control for the parents, and we use the sibling unaffected and who is not a carrier as the control for the younger kids. And we found that the expression of the protein was of the DARS2 mRNA was decreased in the parents as carriers, similarly here in the other sibling who is a carrier, and very decreased in the affected patients. So this is consistent with our presumption and the effect of the gene defect. So we then look at the full protein level via Western blot, and here is the gel results, and here is the quantification based on acting levels. And again, with the normal control for an adult, the normal infant control or child control, and the decreased levels in the carriers and the lower levels in the patients. And there are some fluctuations. All this testing has been repeated multiple times and is consistent with minimal variations. So what was the effect of these mutations in the functionality of the fibroblasts? And we heard before that some mitochondrial diseases cannot be studied in fibroblasts or are difficult to be studied in fibroblasts. And we saw the very interesting presentation of Dr. Van Hoff showing the challenges. And I fully agree with that. There are many conditions in which doing studies in fibroblasts will not be useful. But if and when you have a model of disease or a particular mutation that shows the defect in fibroblasts, it becomes really very important, very valuable, because fibroblasts can be cultured indefinitely, and we can do multiple studies in this particular tissue. Having said that, we are also proceeding to the differentiation of the fibroblasts to iPSCs and neuronal cells, but I don't have those studies to report today. When we did the seahorse studies, we showed that patient 1 and patient 2, here in yellow and green, had lower basal respiration and lower spare maximum respiration, and therefore spare respiratory capacity compared to the control. And this is statistically significant, and all these studies have been replicated several times, and so we are pretty confident on these findings. We were curious to see if lactic acid, which is not increasing plasma in these patients, but is a nice biomarker for the defects in energy production, as it was explained today by Dr. Graneski and others, was increasing the culture media. And so we did find that sibling 1 and sibling 2 had increased production of lactic acid in the culture media, and the differences were statistically significant. This is all normalized to the control. So we have about 140% of the control, either you can say that, or 40% increase, and here about 25% increase. And the technology is always on the right side. We know that in many mitochondrial disorders, the oxidative stress plays a very important role. So, and it's rather difficult to measure oxidative stress, and many different techniques can be done. In this particular case, we replicated a technique that was published for a different disorder. I think was ACAT9 by the group of Jerry Buckley, cited here. And we look at the mitoSOX, which is the mitochondrial superoxide, and we normalize the expression or the amount of superoxide according to the MitoTracker green, which is an indication of the mitochondrial content. So here again, we have the sibling as a normal control, and the two affected patients with some variability, but yet significant differences at a lower statistical value, but significant indeed. We also were curious for the results reported by Lin and others in a recent paper in Taiwan, and then that they showed the morphology in the cells of those two patients was affected, the mitochondrial morphology to be more precise. So we went to the fluorescence microscope, staying with MitoTracker green, and we use deconvolution software with ImageJ plugged in, and everything I'm telling you is what my wonderful technician Winnie tells me that she used. I haven't done any of these. So you see on the left side the sibling 1 and the sibling 2, two pictures of the sibling 1 or control with beautiful cells, beautiful mitochondria, long and networking. And here you see cells of patient 1 and patient 2. You can see the mitochondria are not as elongated, and they have fewer branches and are fragmented. And therefore, you can clearly see that there is a morphological defect in the mitochondria of these patients, which reflects in the microscopic level what you have seen at the functional studies somehow. Now, looking into the microscope is important, but not quantitative. So we went one step further, and then Winnie has used a quantification method called MENA, which stands for mitochondrial network analysis, and was able to document that the footprint, which is the mitochondrial mass, the length of the mitochondrial branches, and the number of branches were statistically significantly decreased, at least for these two parameters, and in one patient for the other parameter, in the two patients compared to the control. And this has been done in multiple experiments, and this particular experiment involves documentation of over 100 cells, which is a lot of work under looking at the microscope. And finally, this graph that you just saw presented by the former speaker, we call it the volcano plot because it looks like a volcano, and this is the lava coming out. And so what it shows here are at the RNA level, which we call the transcriptome analysis. What other RNA from other genes is either overexpressed or underexpressed. And here, as it was shown by the previous speaker, on the left, we have genes that are underexpressed, and on the right, overexpressed. We are in the initial staging of this analysis. We haven't gone through the heat map that was shown before by the previous speaker and also by Dr. Hobart, which we are planning to do. This is just very preliminary data and information to show we are working also in the transcriptome. And we are being very conservative at this point and using a cutoff of 0.1 for statistical significant and twofold decrease or twofold increase in the amount of transcript in the amount of the transcript for each particular gene. So with that, I would like to go to a summary that we found in DARS2 a new novel mutation in our patients. And I would say our results confirm the expression of the disease in fibroblasts, at least I want to emphasize in this particular disease and in this particular mutation, this particular family, we know that may not be possible in all diseases to be expressed well and studied well in fibroblasts. I believe we are expanding on the nature of the mitochondrial dysfunction on LBSL, increasing the knowledge that we have about this particular condition and the way it behaves in fibroblasts. And we plan to use all these metrics for future in vitro therapeutic interventions. I do not have the data here today, but we are working in a couple of different compounds. I can mention one is aminolevulinic acid. The others are in different stages of study and looking to see if we can improve the energy production in the cells. And we continue with the mRNA data. So big thanks to the people working in the laboratory. Winnie is here in the middle. She's the one who has done most of the studies, and missing here in the picture, we didn't have time, is Dr. Alexandra Latini, who is a new addition to our team. She's a PhD expert on BH4 metabolism, and we are planning to work on BH4 pathway and related to mitochondrial disease as well. And finally, I want to thank Research Administration at CHOC. They've been very supportive of us and the families who support our work. And I want to say to the families who are listening here today that it's complicated. you see studies in rats, studies in humans, studies in one disease, studies in one tissue, in yeast, in fibroblasts, and it's very, very complicated, very difficult to put together, and you understand how difficult it is for us to answer your questions. But I in the same way, your commitment, your efforts, and this meeting is a proof of that, are a real inspiration for all of us to continue what we are doing, and hopefully we can provide answers. Thank you.
Thank you so much, Dr. Abdenur. That was a fascinating talk, and I'm really looking forward to the Q&A at the end of our session here to hear more from the audience. So our next session is going to be co-presented by three brilliant scientists. That includes Dr. Amena Smith-Fine, Dr. Christina Mertz, and Dr. Marc Engelen. And so Dr. Smith-Fine is an assistant professor of neurology and developmental medicine. She has a neurogenetics clinic and works in the multidisciplinary leukodystrophy clinic at the Mosher Center for Leukodystrophies. In addition, Dr. Fine is a clinic in the Center for Development and Learning. Dr. Christina Mertz, primary research interest is the application of relevant cell and animal model systems to understand neurogenic and neurodevelopmental disorders. And then finally, Dr. Marc Engelen is a pediatric neurologist at Amsterdam University Medical Centers. And the research program in the department is focused on the clinical and translational research concerning leukodystrophies and neonatal neurology. And I believe Dr. Engelen is going to be up first here. So I will go ahead and turn the floor over to the group for presentations. Thank you so much for your time today.
Thank you very much for the kind introduction. Let me try to share my screen. Let's see. So I hope you can, let me switch to presentation mode. And I might have to switch screens. So I think that everybody can see my slides now. Excellent. Thank you. Well, thank you very much for the opportunity to do a brief presentation on LBSL. Actually, my presentation is going to be really brief and will serve more as an introduction to the next speakers who will go into detail on the biochemistry of LBSL and also about ongoing studies. But I hope to set the stage for them. So, LBSL, caused by mutations in the DARS2 gene, as has already been mentioned, is a relatively new disorder. It was recognized as a distinct entity only in 2003. That, of course, doesn't mean it didn't exist before that, but that was when the typical pattern of clinical features was correlated to a specific MRI pattern and was recognized to represent a distinct entity. And a few years later, in 2007, it was discovered that it was actually one of the RNA synthetase deficiencies caused by mutations in DARS2. And to first describe what typical LBSL looks like as it was originally described, these are some very characteristic MRI images. So LBSL causes a leukodystrophy, so a disorder of the white matter of the brain with a distinct periventricular pattern, but with also recognizable abnormalities in the posterior fossa, both in the brainstem and in the connections to the cerebellum.
Sorry, Mark.
Yeah,
I
sorry.
think we're seeing your title slide still.
Oh, sorry. On my screen, it's actually, I might have. Sorry, let me try again. Let me.
Sorry.
I think it's sharing the wrong screen. Let me just disconnect my other screens. That should solve the problem, I hope.
Mark, in the bottom right-hand corner, you have a few options. You want to select the one that it looks like a TV. It's directly to the negative sign, the minus sign. And if you click that, it'll switch you into presenter mode.
It's frozen.
I think if we can just give Mark just 1 second to reconnect.
And while we're waiting, does anyone have any questions for Dr. Abdenur? I see that he's still on.
Yeah, hi Jose, this is Ali. Since we're still waiting, I actually did have a question. I noticed, I know you said you haven't analyzed the RNA data, but I noticed that there were at least 2, if not 3 other tRNA synthetases that were dysregulated, which is really interesting. And maybe, oh, looks like Mark is back.
Yeah, I'm so sorry. I did not only unplug my screens, but also my network cable. So let me try again. Can you see the slides move now?
Yes.
Oh, great. Okay, so sorry about that. So the typical MRI pattern, and what is unusual for leukodystrophy, or at least not so common, is that there's also prominent abnormalities in the spinal cord, affecting both the dorsal columns and the corticospinal tracts. And this combination is pretty typical. And if you do MR spectroscopy, you find in the white matter that there is a clear lactate peak and not in the gray matter. And this combination is highly suggestive. So what does this cause in the sense of symptoms? Well, because the prominent involvement of tracts going to the cerebellum, there's a coordination disorder, cerebellar ataxia, and because the spinal cord is affected with the long tracts, like the dorsal columns and the corticospinal tracts, it also causes sensory ataxia in the lower limbs and spasticity, causing a prominent gait disorder. And these symptoms occur in the original presentation rather late, in late childhood or even early adulthood, with usually normal cognition and only rarely epileptic seizures. And although the abnormalities are progressive, Dr. Smith-Fine will describe that this progression is generally slow over years or even over decades. But as is often the case, after the original presentation of a disorder, as soon as a genetic diagnosis becomes possible, and now especially with the application of exome sequencing to routine diagnostics, the spectrum expands. And now it is known that this is only part of the story, and there's also much more severe early onset forms of LBSL that are now sometimes diagnosed. Recently, a few years, I think 1 or 2 years ago, there was a publication on a very severe early onset neonatal form, which is devastating, causes massive brain abnormalities with atrophy, and usually causing severe disability and death within the first year of life. So this is really a neonatal ICU presentation. And something in between this neonatal presentation and the typical presentation and an infantile presentation, with a very, very severe leukodystrophy, but absence of abnormalities in the posterior fossa and spinal cords, which you do see in the later onset forms. And these children are usually also very severely affected, more severely so than the typical presentation. So is it understood why this disease spectrum extends also to the some more severe presentations? Well, partly. I think it was mentioned in the previous presentation that many patients with LBSL have a so-called mild mutation, a leaky splice site mutation that allows for some normal protein to be produced. And actually, as far as I know, there's never been description of a patient with 2 mild mutations. So we think that if you have 2 of those mild mutations, you will probably not develop symptoms, or incredibly mild symptoms that are generally not a cause for diagnosis. And apparently, most of the typical onset patients have 1 of these mild mutations and 1 severe mutation. And the few cases of the very severe LBSL tend to have 2 severe mutations. So there does seem to be some genotype-phenotype correlation. And it suggests that even a little bit of normal protein can already rescue the phenotype quite significantly. So this is hopeful for therapy, I suppose, because these splice mutations are possibly could possibly be affected by oligoantisense therapies that have already been applied to other diseases like spinal muscular atrophy. And this suggested if you can even produce a little bit more protein, you could probably also induce a clinically meaningful effect. So that is something to look at in the future. So this was just a very brief overview, and the next presentations will provide much more detail on the very interesting biochemistry and also current research projects at the Kennedy Krieger Institute. So I gladly give the floor to the next speaker, which will be Dr. Mertz, I think.
Yep. Thank you very much, Marc. Great introduction to LBSL and Dr. Abdenur as well. Whenever you're ready to stop sharing. There we go. Okay, thank you very much. Let me know if this is looking all right. You should see my full screen. My name is Christina Mertz. I'm a research scientist at the Kennedy Krieger Institute, and we have some, what I'll be presenting today is basic science research on DARS2 and LBSL. So we all know basically how these enzymes work at this point. What I'm going to talk to you about very briefly is our work on mouse and cell models to better understand the role and the function of DARS2. So I'm going to dive right into that. One of the first things that we did in the lab was to model LBSL using a mouse model. These mice are donated to us very generously from Sandra Trifunovic, who is in Cologne, Germany, and she had created DARS2 flox mice. And these mice have flox sequences surrounding the 3rd exon of DARS2. What we did with these mice is we mated them with CAMK2 alpha Cree animals to generate full deletion, it turned out to be, of DARS2 within these neurons in the hippocampus and the forebrain. And so what we saw with these animals is that they develop normally for the first few months of life, as you can see in this graph here with the control and the mutant mice. What we're looking at is open field. This is the distance that they travel over a 10-minute period. And progressively, we see that the mutant animals, the ones that are lacking DARS2 in these neurons, show a progressive increase in their activity levels. So this was very apparent. If you go down to the animal room, you could just hear them circling day and night. And this was a very significant phenotype that actually prevented us from doing a lot of additional behavioral tests because they were so active. But it was a very apparent phenotype that occurred in all animals with not too much variability, despite this large bar you see here at the end when we start to see attrition of these mice. If you do MRI in these animals around the same time point, what we can see is a progressive loss of loss of brain tissue. At around 6 months of age, these animals show a slight decrease in their brain area. And by 9 months of age, you can see it's about 1/3 of their brain area, which is gone. And that's represented in this ventriculomegaly here, increase of ventricular space. And then also the cortical tissue is significantly decreased. And you see this now as just CSF area. And that's apparent when these brains are extracted from the tissue as well. Further histological analyses of the cortex, this is using methylene blue, shows that we lose specifically in layer 4 of the cortex. Column A is a wild type animal, column B, the 2 columns here are both mutant mice, with zooming in on the cells shown here in wild type, and then here you can see in the B, this is a mutant animal. And what we see is an increase in the number of glial cells and a decrease in the number of nuclei that represent neurons within that cortical area. If we do additional staining of this tissue, we confirm that glial cell presence. This is staining for inflammatory cells. This is IBA1 positive in the green cells. And first, it's very striking the significant difference here in the cortical thickness, which is represented in the graph in B, but also the infiltration of these inflammatory cells within the cortex. So within this area, we see increased number of microglial count, and then we see that the morphology of the microglia within this tissue is also changed. So they're amoeboid, they're activated, and they do express CD68, a marker of activation, which you can see in this image here. So this tissue is from animals that are 9 months of age. And then what we did is we kind of backtracked to see sort of how this progresses. And we did RNA-seq in these animals at 14 weeks of age. So 14 weeks of age precedes a lot of that behavioral change that we see. So these animals are still typically what we consider normal at this point. They don't have that increase in behavioral activity. But what we can see in a subset of these mice, at least, and what I believe if we had done this a couple weeks later would have been all of the animals, is that we see that the inflammatory pathways are already activated. So the red bars here in our mutant mice are representative of increased gene activity. And all of these genes here relate to immune activation. So we have different pathways, cytokine production, inflammatory response, and a lot of other pathways that again reflect significant neuroinflammation. And just to be clear, this was bulk RNA-seq of the cortex within these animals. At the same time, we see decreases in other pathways, and these are all related to synaptic signaling, synaptic organization, and these would, in our mind, represent neuronal loss. So prior to any sort of behavioral onset, we do see that these neurons are affected and that inflammation is significantly increased. And to combat that, we tried a couple different things. And one of those was to treat with a potent anti-inflammatory antioxidant and acetylcysteine. This is a drug that was mentioned earlier today. So we worked with the Center for Nanomedicine here at Johns Hopkins, and they were able to design for us a nanoparticle that allows for delivery to the CNS to inflammatory cells. We treated these animals starting at 8 weeks of age, so even earlier than that RNA-seq was done. And we treated for many weeks, almost 30 weeks. And you can see no effect of that anti-inflammatory in these mice to reduce the behavioral phenotype at the very least. And also the tissue when extracted at this time point was reflective of the knockout vehicle animals. So we did see that decrease in brain tissue regardless of the drug being on board. So just to summarize sort of our mouse model work, we show that neuronal targeting of DARS2 has a significant phenotypic change. And this is contrary to what happens when this same deletion is made within oligodendrocytes. Sandra Trifunovich's group in 2017 published a paper where they used that same mouse in PLP expressing cells. We also tried it in PDGFR alpha expressing cells, and that shows little to no phenotype. So it's really that neuronal phenotype that seems to produce, or sorry, that neuronal deletion that seems to produce the significant phenotype. We show in our mice that the neuronal death is ongoing and severe, and the neuroinflammation precedes the behavioral phenotype. We did try the anti-inflammatory antioxidant, and we also used an ISR inhibitor in these animals starting very early and allowing to go well into the significance of the phenotype, and also so no effect of that drug treatment. So despite that, we were still able to produce an animal model that presents phenotype. And we do believe that this is going to be a useful model for future therapies and then also for testing future therapeutic strategies. So I'm going to jump quickly into some work that we've done with iPSCs. This is work that we've done here at Kennedy Krieger with Dr. Mingyao Ying. He is a researcher here who has developed a rapid protocol for differentiating iPSCs into motor neurons. And we were fortunate to get 7 patients to donate blood, which we harvested peripheral blood mononucleocytes and had these cells reprogrammed at the Cedars-Sinai induced pluripotent core in LA. And we also generated some age and sex matched controls. So these cells we generated, we received back as iPSCs, and then we differentiated them into motor neurons with Dr. Ying's help. This is a quick video just to show the development of these neurons over time. So this is as the neurons are developing as motor neurons past the NPC stage, the neuroprogenitor cell stage. And what we see when we monitor these cells over several days within a high content cell imager is that the arborization of LBSL cells is different as compared to control cells. So the mean branching points and the mean neurite outgrowth is decreased consistently across LBSL cells compared to controls. And so we've done this in a couple different cell lines now, and we see consistent results with the LBSL compared to the control. Similarly, if we look at electrophysiological data, so this is a microelectroarray system from Axion Biosystems measuring spontaneous firing. We see similar findings in that over development or over differentiation, when we consider maturity to be around day 21 in these neurons, we see a reduction in spontaneous firing in the LBSL cells compared to control. So we do see some functional differences within these mature motor neurons. And then touching on mitochondrial activity. So this is looking at a typical profile of the seahorse assay, which Dr. Abdenur has touched on also. This is looking at the oxygen consumption rate in the mitochondrial stress test. And just to orient everybody, what we're looking at here, this is a dotted line of a control. So this is multiple controls that we've averaged together, looking at baseline OCR in this first quadrant, followed by drug treatment with oligomyosin, which allows us to see ATP coupled respiration. FCCP pushes the cells to their maximum respiration rate, and then ritonone, which blocks complexes 1 and 3, and that inhibits all mitochondrial oxygen consumption. And what we like to look at is this difference between the maximum respiration rate and the baseline rate, and that's what we call the spare capacity. So that's sort of the reserve that these cells have in order to respond to additional stressors if necessary. And if we look at what this control looks like compared to LBSL, these are the mature motor neurons. What we see is the LBSL cells have an almost complete lack of response to these different environmental stressors. And importantly, they lose that ability to respond to additional stressors. So that reserve capacity is gone. And if we do this multiple times with multiple different cell lines, we see consistent results with the exception of this case right here. And then a little bit here where you see some reserve capacity, but for the most part, we can see that these neurons are significantly affected in their ability of their mitochondria to respond. So that's kind of our phenotyping, our way to look at the function of these cells and how they behave. And I'm going to switch gears a little bit and talk about protein and some gene expression experiments that we did. This is studying the DARS2 protein, so just simple Western blots of control iPSCs and neurons compared to LBSL. So this is 3 different patient lines in their iPSCs and neurons. And the first thing that we noticed is that in a control situation at the iPSC stage versus the neuron stage, we see that in the control situation, there seems to be reduced amounts of DARS2 that are necessary, perhaps just for regular function. And then if we look at LBSL in the iPSC stage, we see a reduction compared to control. And then when we look at the neurons in LBSL, we see almost a complete lack of a band there. So despite the fact that there's reduced DARS2 in neurons compared to iPSCs, LBSL is always lower compared to control. So this is just looking at DARS2 protein total. What we also did is we isolated mitochondria from these cells and ran proteomics on what we call the mitochondrial enriched fraction. So obviously these are not all mitochondrial encoded proteins, but these are, or mitochondrial produced proteins, but these are proteins that are associated with the mitochondria when we do the enrichment. And we see a significant number of upregulated genes. We've seen this before in a couple other presentations with sort of this bias towards upregulated versus downregulated in these volcano plots. And then another thing that we wanted to analyze here is the substitutions from aspartic acid. So if we think that DARS2 is responsible for attaching aspartic acid and putting aspartic acid as part of that polypeptide chain, we would assume that perhaps if that role is not being filled, that those amino acids are switched for other amino acids. And if we look at the substitution rate from aspartate, what we see is that's not really the case. So if we compare, let's see, methionine, we see there is not an increased number of methionine substituted for aspartate. So we sort of assume here that the fidelity or that the ability of DARS2 to link aspartic acid as part of that polypeptide chain is still happening as it should be. If we look specifically at mitochondrial encoded proteins, we were able to detect a few within our samples. This is a control versus 3 different LVSL patient lines. And we do see significant reduction in a few of these mitochondrial proteins compared to control. If we look at the percent of aspartic acid within these proteins, we don't see a relationship. So again, with the same logic as previously, we would assume that the higher the aspartic acid content, the more dysregulated that protein is, and that doesn't seem to be the case. ND2, for example, does not contain a single aspartic acid, and it is reduced by 50%. So there does seem to be some other factor at play here, which affects mitochondrial protein production. Looking at a gene ontology chart, just of the protein changes as a summary, what we see is the majority of these proteins that are affected have to do with protein binding, have to do with DNA binding. And if we look at another hierarchical plot here, we see that a lot of it has to do with regulation of splicing, regulation of mRNA processing, and these sorts of activities. So again, not things that are traditionally related to straight up translation. So just to chase this down a little bit further with another in-depth look, a final sort of approach that I'm going to talk about is the use of LV-cell cerebral organoids. So this is work done by Shritzi Guang. She was a medical student in the lab for 2 years, and she sort of spearheaded this project to differentiate iPSCs using an unguided protocol into cerebral organoids. I'd like to emphasize that this is preliminary data. The controls that we used here were other patients or other samples that we were able to purchase from Cedars-Sinai. So these are not isogenic controls. This is all preliminary unpublished data, but we do think it's interesting to share with you. These organoids were, this is a day 21 organoid. We actually allowed these to grow until day 70, and then we harvested them. We used an approach called SmartSeq2. SmartSeq2 is a very high resolution method for single cell RNA-seq. So this does full length and even reads across transcripts, and it's really good at looking at transcript level data and to get alternative splice forms. The cost of that resolution is that you can't look at a lot of cells. So across 10 samples, we did 196 well plate per sample. And then after quality control, we were left with 809 cells that are used for analysis. You can see in this plot on the right, this is a distribution of our patients. So you can see that everyone is sort of mixed in well, which is what we would want to see. And if we categorize these cell types or figure out what kind of cell types they are, you can see that we got a variety of cells, including glial cells, cortical neurons, radioglial cells, and neuroepithelial cells. So we have a lot of data on this, and I'm just going to show you a little bit, but one of the most interesting things that pulled out is when we looked at our LV-cell patients, it seemed like our LV-cell patients sort of clustered into 2 groups, which we were not expecting. And these 2 groups had unique profiles, and you can see in group 1, we have a decrease in RNA splicing. In group 2, we actually have an increase in RNA splicing. And then group 1 had increases in pathways relating to cell death, programmed cell death, and group 2 seemed to have more increases in pathways related to mitochondrial function, so OXPHOS, ATP synthesis, respiratory chain complex assembly. So we definitely thought that these sort of differences between the 2 groups were very interesting. And further exploration into this led us to realize that the expression patterns seem to relate to the mutation type. So they seem to cluster based on what type of mutation they have. So this is the DARS2 gene, and these are looking at the mutations of the patient lines that we have. So we have 4 lines that contain either 2 missense mutations or 1 splice site and 1 missense mutation. And then we have 3 patients that have 2 splice site mutations alone and no missense mutation. So it seems like the pattern that was generated is based on what we say is the presence of a missense mutation, which is very interesting. And obviously with a small group, it's hard to do any sort of phenotype genotype correlation, but this is the first time that we've seen any sort of separation of the data like this. So just looking a little bit more closely into these data, this is looking at neural epithelial cells. The dots that you see here, the size relate to the percent expression, and the color relates to the log2, the fold change. So this is looking at control levels. If we look at the group 2, so this is those patients with the, just to be sure I'm saying this right, the frame shift, or sorry, the 2 splice site mutations. They resemble controls very well. And if we look at the group 1, so that's containing a missense mutation, we see that they are decreased compared to both group 2 and control. A similar pattern is observed with our cortical neurons. So group 2 more closely resembles controls, and those patients with the missense mutations have decreased both expression and fold change of these genes related to splicing. We look at endoplasmic reticulum cholesterol synthesis. This is another pathway that came out. Again, we see in radioglia, group 2 and controls resemble each other, and group 1 have increased expression of these genes, and the similar pattern for cortical neurons. And then finally, if we look at genes related to translation, here we see the opposite effect, where group 1, so those missense mutation patients, closely resemble controls, while the group 2 show increased activation of these genes relating to translation. And the same is also true for cortical neurons. So this is, again, very preliminary data. These are controls that are not isogenic. So this is something that we're going to chase down a little bit further, but we think is a very interesting start. So as I don't need to reiterate, the tRNA synthetase mutations present as very complex disorders. In my mind, it's likely that the effects of these mutations extend beyond amino isolation. And we have been able to show deficits in growth and projection, spontaneous firing in these motor neurons, and also effects on the mitochondrial function. The transcriptomic analysis reveal unique signatures that may correspond to specific mutations, which might lead us down the road to some more information about mechanism. And as always, much work is left to do, so we hope meetings like this continue in the future, and we'll be able to share some updates in the near future. So with that, I'll finish and let Dr. Smith-Fine go. Just a quick thanks to the lab here and our considerable support staff. And of course, funding. We work very closely with the Cure for Ellie, and they've been wonderful in providing funding for us for the last few years. So with that, I will stop share. Thank you very much.
Thanks, Dr. Mertz. And I will attempt to share my slides. Can you see the full slide presentation?
Looks good.
Okay, great. All right, so thanks very much, Desiree and Ashley, for the invitation to speak today, and giving us this opportunity to highlight our translational LBSL research efforts at Kennedy Krieger. So this afternoon, I'll basically give you an overview of our ongoing natural history study in LBSL, in which we're using wearable sensors to gather quantitative information about gait and balance through virtual visits in patients' homes. All right. So I'll start by outlining our questions and goals for research of the natural history of LBSL. And I know you've already heard from our previous speakers and Marc Engelen most recently about a little bit about the clinical phenotype in LBSL. So, of course, the motor impairment is really the major feature that's common sort of across the spectrum with ataxia and a progression to loss of walking or loss of independent walking being kind of one of the major quality of life factors. So in terms of the motor impairment prognosis, we know that half of patients require walking aid by adulthood, 13% become fully wheelchair dependent, and so far we know that earlier age of onset is associated with sooner loss of independent walking and wheelchair dependence. But beyond the age of onset, it's really unclear still which factors determine the rate of disease progression for an individual. So some thoughts, this certainly may in part be related to genotype, but the genotype-phenotype correlations has been somewhat limited because this is a rare disorder and there's a high number of private mutations, as you know. Also, the progression rate could be related to the white matter lesion burden or anatomic location. And this has actually been, there's some nice publications investigating this. And in general, more extensive white matter involvement has been associated with greater motor impairment in LBSL, but there are some patients that have really extensive MRI lesions and yet are still asymptomatic. And then it also sort of paradoxically appears that the T2 lesion hyperintensity improves over time despite clinical deterioration. It's only a part of the picture. So at this time, there are no FDA-approved treatments, although as Dr. Mertz and others have mentioned, there are multiple potential therapeutic compounds in the translational pipeline, which is very exciting. And so at this point, though, the lack of longitudinal outcome measures that represent clinically meaningful measures of disease progression is one of the unmet needs we need to address to be able to advance to the clinical trials phase. So with this in mind, our main study objective is to establish clinically meaningful quantitative performance measures for trials in LBSL. The approach we're using is to gather information on gait and balance using wearable sensors and basically accelerometers and an ataxia scale. We're also getting clinical history and neuro exam information from patients. So we perform assessments during virtual visits using Zoom, actually at participants' homes. And this has really been great for increasing participation and decreasing the burden of travel for our patients. Obviously, also a big advantage in the pandemic era when travel has also been unsafe. And we're testing both LBSL patients and age and sex matched healthy volunteers. And these assessments we've been repeating every 6 months. They will be for a total of 5 years. So we chose the OPAL system of wearable triaxial accelerometers for our study. It's a non-invasive, relatively inexpensive technology for recording movement in a real-world context. The OPALs have been used to measure disease-specific features in other neurodiseases like multiple sclerosis, Parkinson's disease, and cerebral palsy. And then similar metrics of gait and sway have been reported in other disorders that feature ataxia. So the system consists of 3 watch-size battery-based accelerometers, basically looks like a Fitbit, and these send a synchronized signal to a sensor through ultra-low power radio frequency waves. So the 3 sensors are placed on the participant's feet and waist for a series of brief walking and standing balance tests. And so I'm just showing for the virtual visit home. At home, we send the OPALS kit, a webcam, a laptop, and a device manual that gives all the instructions needed to set it up. And here's an example of one of our patients, Ellie McGinn, and her mother participating in this research. Okay. So I mentioned our clinical ataxia scale. So this is a brief sensitive tool for assessing ataxia. It's been used in other ataxic disorders. And it's had the advantage of showing progression in spinocerebellar ataxia after a year, but wasn't able to capture all ataxia symptoms reliably at disease onset. And so basically that's a limitation. But wearable accelerometer measurement of gait ataxia was shown to be superior to the SARA in the early stages of spinocerebellar ataxia. So we're basically exploring the use of both of these measures for assessment of disease severity and progression in LBSL. Here's one of our coordinators demonstrating this test. Okay. So within we have a larger group of about 20 patients enrolled in our natural history study, but there's a group of 8 patients who are able to walk and follow instructions consistently for these tests, and they've been enrolled since the very beginning, and we have measures for at least 2.5 years. So I'll focus on that group. And the median age range, these are all really young people. The median age is 10 years with a range of 5 to 23 years, roughly 40% females, 60% males. The mean age of onset was 4.5 years with a range from infancy to adolescent onset of symptoms. And the presenting signs are ataxia and fine motor delay really universally. 75% of our participants reported a brief worsening of motor skills and sometimes cognitive sluggishness after a fever, illness, or minor head injury that usually resolved after a few days to a week. That hadn't been reported in LBSL yet to our knowledge, but that is a pretty common thing to see in mitochondrial diseases. And then just to give a sense, the gross motor function classification scale, or the GMFCS scores were relatively low, 1 to 2 on a scale of 1 to 5. And then the max, which looks at manual ability, hand ability, was also in that low range of 1 to 2 on a scale of 5. And then the SARA ataxia scale, mean was 8 with a range of 1 to 16. So this is really mild to moderate clinical ataxia. And just further breaking down that ataxia scale, most participants had signs of ataxia affecting their legs, followed by ataxia in the arms and trunk, and only about 40% had mild speech impairment. Okay, so transitioning to the OPALS testing. For the 2-minute walk tests, participants just walk back and forth between 2 points on a 22-foot walkway set up on a clear path in their home for 2 trials. Just for reference, a gait cycle is the period from the foot's first contact with the ground, typically a heel strike, until the same foot makes contact with the ground again. And these sensors collect 16 variables during each gait cycle, which really encompass all different phases of where the foot is positioned in space during walking. So this is a 10-year-old girl with a moderate SARA score. Just to give you a view of what this looks like in the home. So notice some mild stumbling with her gait and hesitancy with turns. Okay. So the stride length is the forward distance traveled by the foot during a gait cycle. And a short stride length is seen as a compensation basically for unsteady gait. It's commonly reported in ataxia. So as we hypothesize, our LBSL group did demonstrate a shorter stride length compared to age-matched controls, although this wasn't a statistically significant difference in our small group of participants. The time it took to complete a gait cycle in seconds was slightly longer for the LBSL group, and then overall, sort of putting these variables together, they had a slower stride velocity or gait speed compared to the control group. And in the literature, difference of this degree of about 0.1 meters per second is clinically meaningful. So the hallmark of ataxia is course variability, which can be estimated by this lateral step variability marker. So when considering 3 consecutive foot placements made by the same foot, this describes the variability of the perpendicular deviations of the middle foot placement from the line connecting the first and third. So essentially, this indicates a varying or stumbling gait, and larger deviations might indicate a wider stance base to preserve stability. So our LBSL group showed a larger lateral step variability versus controls by about 2 centimeters. And there's also quite a larger range of values for this group. So circumduction is a lateral swing max, or the maximum amount the foot travels perpendicular to forward movement during a stride. Positive values indicate more movement to the outside. And this is a gait feature seen with leg weakness or increased tone in the legs or ankles. So our LBSL participants had a wide range in circumduction and slightly greater average than the controls. And then we also looked at the height of the foot at mid-swing relative to its start position while standing, and found a statistically significant difference in the average for between controls and LBSL. And so this elevated step at mid-swing is also called a high steppage gait, and it's been reported in disorders featuring cerebellar ataxia, peripheral neuropathy, or spinal cord lesions. This high step basically is a compensation to clear the foot of the floor during stride. So the remote balance testing consisted of 4 postural sway conditions in which we challenge participants by either removing visual input or narrowing their base of support. They do 2 trials of each condition. Their stance width is standardized with a foot plate, and sway is measured from a sensor on the waist. So these clips are representative of LBSL patient performance on the balance tests. First, feet apart, eyes open. Feet apart, eyes closed.
Almost done.
Which is more challenging.
10 seconds left.
Feet together, eyes open. And then feet together, eyes closed, which is very difficult for some of our patients to complete. And so above are plots mapping this girl's sway path compared to an age-matched healthy volunteer. This is looking basically at tracing the path of the sway over 30 seconds from the top down. So across all conditions, you can see easily there's a greater sway for our LBSL participant, but especially in conditions when her eyes are closed. And it's greatest when her eyes are closed with a narrow base of support. So here are the summary sway plots for the control and LBSL groups. So as in our representative girl, the sway area is greatest and the feet apart, eyes closed, and the feet together, eyes closed conditions with a quantification below. And then to further evaluate the impact of removing visual input on sway, Romberg quotient was calculated for each patient. And this is the Romberg quotient is the sway area with the eyes closed divided by the sway area with the eyes open. So we saw that the average Romberg quotient was significantly increased for LBSL subjects, which we think likely reflects impaired proprioceptive contributions of the dorsal column pathway fibers in the spinal cord. So the thought is that the myelopathy seen in LBSL might really be an important contributor to the overall motor disability experienced by these patients. So lastly, these two plots demonstrate that the SARA ataxia score is strongly correlated with lateral step variability score in walking and the sway area during sway, supporting the idea that these sensors are capturing variables that are clinically meaningful and represent ataxia. So in summary, through virtual testing at home, we found LBSL patients walk and turn slower, have shorter stride lengths, and show more gait variability compared to healthy volunteers. Removing visual input has a greater impact on the sway area than narrowing the stance base, suggesting that dorsal column dysfunction is an important contributor to instability while standing. And the step variability in sway area measured by the wearables is strongly correlated with a well-known clinical ataxia scale. So this likely reflects a relation to sensory or cerebellar ataxia. So I want to end the last several slides by showing a few key variables from our 2.5-year follow-up visits. So when interpreting this long-term data, it is important to consider that for children and young adults, the normal values of walking change over time with age and presumably with height. So as examples, both the stride length and the gait speed increase with age, and these changes are really steady and incremental every couple of years. And then on the other hand, the cadence in steps per minute decreases consistently with age, which makes sense since the strides are actually getting longer over time. So at 2.5-year follow-up compared to Opal's walking values from a large healthy control database that was recently published, we saw that the gait speed for our LBSL participants actually dropped, which is exactly the opposite of what's expected for healthy young adults. Double support is expected to stay relatively stable around 16 to 17% for controls throughout childhood and adolescence. And this, the double support is the percentage of time during the gait cycle that both feet are on the ground, with high numbers indicating more gait instability. So we saw for LBSL patients that the time spent in double support started a little higher at 18% and then increased further to around 20% after a couple of years. The stride length did increase slightly in LBSL as expected, but not to the same extent as in healthy controls. And then finally, the cadence and steps per minute did drop in LBSL to a greater extent than expected for healthy controls over time. So with this early sort of snapshot of the data, we see that multiple gait parameters are diverging for LBSL participants compared with expected control values over 2.5 years. And I think most notable are that the gait speed is decreasing and the double support is increasing, which is opposite the trend we would expect with increasing age. So these are just individual patient line graphs for the plots I just showed. So the warm colors, pink, red, and orange, represent 3 patients that showed a consistent pattern of changing gait stability. So if you sort of look across the figures, these 3 have a slower gait speed, increased time spent in double support, a decreased cadence, and a shorter stride length. And so for the other participants denoted in cool colors, the follow-up values were relatively stable or slightly improved in a direction that was expected for age. So postural sway, or the standing balance, is also a dynamic process with aging, making it more challenging to evaluate what's actually a pathologic change in balance in young people with LBSL. So a large database of healthy control sway values in children using the opals was published in 2021, which is very useful in interpreting our own data. So first, from this paper, we see there are expected declines in sway area values with age, especially in the eyes closed conditions. And this is like a dramatic drop between about 5 and 10 years of age. So looking at our data at 2.5 years, we found the sway values, they did remain relatively elevated for LBSL patients compared with controls in this feet apart, eyes closed condition. But they still showed sway over a smaller area as they aged. So essentially it'll be critical in the next steps of analysis to include modeling that accounts for age. So our next steps basically are to determine the usefulness of opals remote testing over time, over the full period of 5 years. And we are continuing, we actually began a collaboration with Dr. Marc Engelen, who previously spoke, and Dr. van der Nap at the UMC Amsterdam to recruit European LBSL patients. So of course, since Dr. van der Nap first described the disease, she's familiar with a large group of patients there, and we're delighted that they've joined with us. They have opals kits for use and have been trained, and I believe they started in summer 2021 with recruitment of adult LBSL patients. So with that, I just thanks again for the opportunity to speak. We're very grateful to our LBSL participants and families who have been hanging in with us at this point over 3 years. And a great thanks to the Fatimi Lab and our colleagues there, and the A Cure for Ellie Foundation and the McGinn family, who has supported this work financially and in many other ways over the last few years. And just one quick plug, we are in the planning stages of a LBSL Family and Scientific Conference in summer 2022. We hope it will be hybrid, although the virus is unpredictable, so possibly switch to all remote. And so in addition to talks, we'll be offering clinical visits and research visits on site as well for those who participate. Thank you very much.
To those affected by mitochondrial disorders, UMDF is here for you. Since 1996, we've worked to provide support, fund research, and create awareness.
Our mission is to promote research for cures and treatments of these diseases and to give support to affected families.
We want future generations to grow up in a world without mitochondrial disease.
I'm playing for the United Mitochondrial Disease Foundation.
Look at that. Nice check for you. Over the years, our community has grown and our message has sparked a movement.
Hey everybody, it's Maya Mialik. Have you ever heard of mitochondrial disease?
Together, we've been able to make a difference for patient families around the world. Today, we are helping thousands of people navigate their mitochondrial disease journey. We're here, listening and providing support. We're here, educating and connecting patients and healthcare providers. We're here, finding and funding the best cutting-edge research. We will not rest until we find a cure. Join the movement at umdf.org.
Hi everyone, I'm Kyra Mann, CEO of MitoAction, and I'm honored to be here today to support Cure-aaRS and the first annual Mitochondrial ARS Gene Scientific Symposium. Thank you to the Cure-aaRS team for giving me an opportunity to introduce you all to MitoAction. MitoAction is a patient advocacy organization that was founded in 2005 to support families affected by mitochondrial disease. Through support, education, and advocacy, MitoAction is committed to walking with our families on their journey with Mito, advancing therapy development, and hopefully one day a cure. Through our programs, such as our weekly support calls, Mito Socials, and the Mito 411 support line, we are here to answer your questions, connect you to resources, and sometimes just to be a listening ear so you know you're not alone. Our Matthew Hardy Camper Fund and Scholarship provides kids with mitochondrial disease the opportunity to attend summer camp and provides financial support for college students with Mito to fulfill their dreams of obtaining a college degree. New this year, we launched a partnership with Give Kids the World Village to provide our families with once-in-a-lifetime wish trips to the village in Orlando, Florida. The MitoAction mobile platform, MyMito, helps families and their loved ones track their day-to-day journey with Mito, complete with symptom tracking, medication tracking, incident reporting, scheduling, telehealth, medical record storage, and more. We are excited that we have just launched a partnership with the CPATH Institute to provide, with user consent, de-identified data from the platform to help researchers and clinicians better understand mitochondrial disease and the challenges our community faces on their quest for therapies and ultimately a cure. Each month, MitoAction brings leading clinicians, researchers, and medical professionals to share the latest information in mitochondrial medicine in our monthly Mito Expert Series. Our podcast, Energy in Action, gives a look into the lives of families affected by mitochondrial disease, shares their triumphs and stories of inspiration as they live a full life despite their diagnosis. This is just a sample of the programs and services that MitoAction provides to the community. We encourage you to reach out to us, get involved, and participate in the programs that are most meaningful to you on your journey. To learn more about MitoAction, visit our website at www.mitoaction.org. We look forward to connecting with you, walking with you on this journey, and supporting you in the ways you need us to be there for. Thank you to all of the clinicians and researchers who have given their time for today's conference. Because of you, our families can truly believe that hope is on the horizon. Thank you and enjoy the rest of the conference.